Hello students, I'm Dr. Shivika Sethi, and in today's video I'm going to be discussing the microbiology MCQs that were asked in the INI-CET November 2024 exam. Now since these questions are recall based, there might be some discrepancy in the question or in the options, but we have tried to be as authentic as possible, and that's the reason perhaps we've taken a bit of time to put up these lectures, these discussions. So before coming on to the actual questions, let me just quickly go through what were the topics that were asked in the exam. So we had two questions from sterilization and disinfection. One of them I will be discussing, the other one will be taken up by the PSM faculty. Then we had a question each on arrangement of bacteria and morphology of bacteria. So this is basically general bacteriology. Again, a question from general bacteriology, culture medium, antibiotic sensitivity. So I always say that in INI-CET exams, sterilization and disinfection is a very important topic, and general microbiology or general bacteriology are very, very important topics. Then we got a bouncer in the form of 4 diseases, their etiological agents, we had to match them. Then how do hepatitis causing viruses transmit? Then we had questions on vaccine preventable cancers. Then 2 questions from trauma-acquired infections, then 2 questions from diarrheal agents, 1 question from etiological agents of urethritis, then Cryptococcus, we got a big question, Ascaris, another big question. Then we got an image-based question on Echinococcus, that is Hydatid disease. Diagnosis of malaria, however, this question is going to be taken up by the PSM faculty. Then we got a question on flukes. This is basically a question regarding treatment of flukes, and this will be taken up by the pharmacology faculty. Okay. Then we got 2 questions from immunity. Right. So let's get cracking with the questions. So our first question was, bacteriological loops used for culture media inoculation are sterilized by which of the following methods? Autoclave, placing in flame 3 times, placing in flame till it becomes red hot, and microwave. Now this is a very, very easy question. If you worked in a microbiology lab during your second year, you would always remember that you put your bacteriological loop in the flame, make it red hot, and then sterilize it. That's how you sterilize it, by making it red hot, right? So that is definitely our answer, right? You can, yes, autoclave them, but do you know how often we need to sterilize the inoculation loop? Repeatedly, every culture medium. You cannot just bank on the autoclave, getting it autoclave, and you cannot have hundreds of microbiological loops, because these are pretty expensive. Either they are made of platinum, that would make them very expensive, or sometimes they are made of nichrome, relatively cheap. But yes, you can autoclave them, but most often they are sterilized by placing them in flame and making them red hot. Okay, that's how we do them. Okay, let's go back to these options. What would you place in flame 3 times? Just passing them through the flame 3 times. That is what we do for disinfection of your slides and cover slips. Let's move on. A 20-year-old develops a pustule progressed quickly to necrotizing fasciitis. Gram-positive cocci in chains were seen on microscopy of the exudate. Which of the following is the most likely etiology? So here this is the biggest hint to us. Gram-positive cocci in chains. Amongst the options, Streptococcus, Enterococcus, Staphylococcus, and Micrococcus. This is very easily a straightforward answer. And anyway, we know that the most common cause of necrotizing fasciitis is Streptococcus pyogenes, or the group A beta hemolytic Streptococcus. Staphylococcus would be arranged as gram-positive cocci in clusters. Micrococcus would be arranged as gram-positive cocci in tetrads, that is sets of 4s. Enterococcus would be arranged in pairs, generally pairs at an angle to each other. Moving on, a farmer comes with a wound acquired a couple of weeks back following a blunt trauma from a farming tool. Microscopy of the wound exudate showed weakly acid-fast beaded filamentous rods as shown in this figure. What is the most likely etiology of this infection? Nocardia, Bacillus anthracis, Listeria, and Mycobacterium tuberculosis. Now, you can see in this image, they have mentioned weekly acid fast. That means this is the acid fast stain, right? We can see those red colored rods against a blue background, that is the counter stain. So, these are what is the morphology? These are filamentous rods that is mentioned out there. And whenever we read the word filamentous rods which are showing branching, we are going to think of the family Actinomycetes, the family Actinomycetes. Okay, these are, this is a big family containing many genera under it, and all of them morphologically are gram-positive rods which are showing branching, and they are very, very slender or filamentous. That's what we call them. Okay. Now, amongst in this family, we have two important members here which are clinically most important for us to remember: Nocardia and Actinomyces. Don't confuse Actinomyces with the family Actinomycetes. Okay. Now, Actinomyces and Nocardia morphologically on gram stain would be similar, but Nocardia, this is weakly acid fast or partially acid fast, meaning acid fast with a lower concentration, 0.5 to 1% of sulfuric acid as the decolorizer, whereas Actinomyces are not acid fast. Another important difference to remember is that Nocardia are aerobic, whereas Actinomyces are anaerobic. And Nocardia, these are always exogenously acquired infections because these are normal environmental saprophytes, whereas Actinomyces are always endogenous infections because these are present as normal flora in our oral cavity, in our GI tract, or in the female vagina. Okay. So, this infection is generally endogenously acquired. So, let's go back to our question. Very easy. Nocardia is our answer. Bacillus anthracis is also a gram-positive rod, but it is not having this kind of slender morphology, and it does not show branching, and it is not acid fast. Listeria is also a gram-positive rod, but not acid fast, not branching, not filamentous. MTB is definitely acid fast. I won't say it is weakly acid fast. It is acid fast with 20% of sulfuric acid, but it does not have this kind of morphology. Okay, let's move on to our next question. Which culture medium is used for the isolation of Mycobacterium tuberculosis? The easiest question that you can be asked for isolation of M. tuberculosis is Lowenstein-Jensen's medium, an egg-containing medium. It's a green medium which is dispensed in a bottle. You must have seen this McCartney's bottle. And what is the selective agent out there? What is giving it that green color? And that acts as a selective agent inhibiting the growth of unwanted bacteria. That is malachite green. Right, that's our McCartney bottle containing the LJ medium. How long do MTB take to form colonies on this medium? Because of the long generation time of 14 to 15 hours, they will form colonies in 3 to 8 weeks. And what is the colony morphology described as? Rough, tough, and buff. Buff meaning off-white. Rough, tough, and buff colonies. Okay, let's move on. Which of the following is a test for carrying out antibiotic sensitivity testing? CAMP test, E-test, latex agglutination, and the Elek test. That's very easy. If you've done general bacteriology, the epsilometer test, or in short, the E-test, is what is used for antibiotic sensitivity testing. It looks like this, right? So we have our Muller-Hinton-Agar plate, on which we have these plastic strips which are impregnated with the graded concentration of antibiotic. So after preparing a lawn culture of the bacterium, swab inoculating the colonies, you just place these plastic strips, and after overnight incubation, you will get these kinds of ellipsoidal zones of inhibition. And where this zone of inhibition intersects this plastic strip, that gives us what is the minimum inhibitory concentration of the antibiotic for this particular bacterium, right? So the advantage of the E test is it determines the minimum inhibitory concentration. But these are expensive tests. In our country, it is difficult to acquire these because of, restrained resources. So instead, what we have is, what we most commonly use is the Kirby-Bohr disc diffusion test. Kirby-Bohr disc diffusion test. So we have here discs which are impregnated with a single standardized concentration of the antibiotic, and after overnight incubation you will see zones of inhibition. You measure the diameter of the zone of inhibition and you say it is sensitive, intermediate sensitive or resistant to the antibiotic depending upon the diameter of the zone of inhibition. So what is the disadvantage of the Kirby-Bohr diffusion test? It is not determining the minimum inhibitory concentration of the antibiotic. Exact concentration cannot be in. So this is just a qualitative test. It does not determine the MIC, but it is very, very cheap, very, very easy to do, right? So unlike the the E test, which is a quantitative test, it exactly quantifies the minimum inhibitory concentration, okay? So the other options that were mentioned in the question out here, these are other tests commonly used in bacteriology. This is the CAMP test, Christie, Atkins, Munch, Peterson test, which is used for the identification of group B beta hemolytic streptococcus, also known as Streptococcus A galactiae. So here we have Staph aureus, vertical streak, horizontal streak is of group B Streptococcus, and we see these enhanced zones of beta hemolysis, also called as arrowhead hemolysis, because their hemolysins synergize with each other to produce these arrowhead hemolysis. The same CAMP test is also positive for Listeria monocytogenes. Okay, so CAMP test is positive for both these bacteria. Next test that was mentioned out here in the option was Elek's gel precipitation test. This is a test which is used for determining the toxigenicity of Corynebacterium diphtheriae. So we have a filter paper strip which is placed on the medium when it is liquid. It's not yet absolutely solidified. This is impregnated with the diphtheria toxin. At right angles, we are going to streak a known toxigenic strain and a known non-toxigenic strain of Corynebacterium diphtheriae, and in between our test isolate. And after overnight incubation, the toxigenic strain of Corynebacterium diphtheriae will secrete the diphtheria toxin, and it will produce these kinds of lines of precipitate. And if our strain is also producing the diphtheria toxin, we will also see similar lines of precipitate, right? So this is what kind of a test. If you go back to the antigen antibody reactions, it is a double diffusion in 2 dimensions test. Double diffusion in 2 dimensions, also called as the Ochter-Loney's test. So both toxin and antitoxin, when they bind to each other, they form these kinds of precipitate. Okay, last the option that was mentioned out here was the latex agglutination test. Latex particles are coated with antibodies, and these can be mixed with the specimen of the patient, and we can use it for detecting any kind of antigen. Just coat the latex particle with the antibodies against that particular antigen. So you can use it for many, many tests. An example is detecting the capsular antigen of Cryptococcus in CSF or blood by coating the latex particles with antibodies against the capsular antigen of Cryptococcus. Right, next, moving on, match the disease with the etiological agents. So donovenosis, we know we study about it in genital ulcer disease, so we can never afford to forget what is the cause of donovenosis, also called as granuloma inguinale. Genital ulcer disease is a very, very important disease. So we always should remember that the causative agent is Klebsiella granulomatis, earlier called as Calymatobacterium granulomatis. Oroya fever is a disease which is reported mainly from South America. It is also called as the carrions disease, transmitted by the bite of a sandfly, and it is caused by Bartonella bacilliformis. Rat bite fever, the cause is Streptobacillus moniliformis, and Pinta is caused by Treponema caretium. Okay. Remember that rat bite fever is also caused by Spirillum minus. So in the Western world, it's caused by Streptobacillus moniliformis, but in Asia, etc., so rat bite fever is caused by Spirillum minus. Which of the following is primarily a waterborne disease? Hepatitis A, B, C, and D. We know there are 5 hepatitis-causing viruses, out of which A and E, these are vowels. A and E, remember, hepatitis with a vowel comes from the bowel. So these are waterborne. The rest of them are transmitted by other routes, right? So this is a very easy way to remember. So obviously, what is our answer? Hepatitis A. Which of the following cancers can be prevented by vaccination? Cervical, Burkitt's lymphoma, Merkel cell lymphoma, and hepatocellular carcinoma. So the answer to this is cervical cancer can be prevented, hepatocellular carcinoma can be prevented by giving their respective vaccines. The answer to this is option C. Cervical cancer is caused by human papilloma viruses. There are more than 150 human papilloma viruses. However, the high-risk ones have the propensity to cause cancers, and we have a very nice vaccine against it, which is marketed by the name of Gardasil. Okay. So Gardasil, earlier we had the quadrivalent vaccine, which contained the major capsid protein. Remember, this is a very commonly asked question, major capsid protein of human papilloma viruses. What is the name of this? L1 protein. Major capsid protein, which is synthesized by recombinant techniques, and the Gardasil one, the quadrivalent one, contained the L1 protein of 6, 11, 16, and 18. And in recent years, we have Gardasil 9, which provides added protection against 31, 33, 45, 52, and 58, apart from these 4. Okay. So remember the L1, the major capsid protein of these genotypes of human papilloma viruses. Of course, we know that for hepatocellular carcinoma, hepatitis B vaccine, if it is administered, it can prevent cancers. Okay. Hepatocellular carcinoma. What does this vaccine contain? It contains the Australia antigen or the HBS antigen or the surface antigen, which has been synthesized by recombinant methods. Merkel cell carcinoma is caused by polyoma viruses. We don't have any vaccine against it. And Burkitt's lymphoma is caused by Epstein-Barr virus. We do not have any vaccine for Epstein-Barr yet. So our next question is, a gardener who is a resident of the sub-Himalayan belt presents with plainless subcutaneous nodules and shallow ulcers on the arm. Axillary lymph nodes are enlarged. Aspirate from the lymph nodes show elongated yeasts, which are 3 to 5 microns. Culture on fungal medium was done. The image of the colony isolated at 22 degrees Celsius. The LPCB mount is shown. What is the likely diagnosis? Sporotrichosis, chromoblastomycosis, coccidioidomycosis, and histoplasmosis. Now, this is a very, very easy question. A rose gardener or a gardener from the sub-Himalayan belt has presented with sporotrichosis, right? So we are seeing it's a dimorphic fungus. We are seeing yeast in the biopsy, and when we grow it at 22 degrees Celsius, it converts into the mold form. And what is the LPCB mount showing? It is showing hyaline septate hyphae with bunches of conidia. We call them as rosettes of conidia. right? So dimorphic fungus which is acquired by trauma is Sporothrix shenkai. Our answer is option A, right? So Sporothrix shenkai, those are other elongated yeasts. In tissue specimens, they are described as cigar-shaped yeast. And that's our rosettes of conidia arising from hyaline septate hyphae. A farmer presents with swelling of the foot. He gives a history of always walking barefoot. Now, there is some issue, some doubt regarding the actual wording of the question. On examination, there are multiple ulcers or sinuses. I do not know what was mentioned out here. Which of the following infection could have been acquired by the farming due to walking barefoot? Mycetoma, coccidioides, Aspergillus fumigatus, and Curvularia. Very easy. This is mycetoma or mother of foot. There is swelling of the foot. There are multiple sinuses. Okay, so this can be easily acquired barefoot. So mycetoma, you must be remembering there are two types of mycetoma, actinomycetoma and eumycetoma. When it is caused by a bacterium which are filamentous branching like nocardia, it is called as actinomycetoma. When it is a fungal cause, it is caused as eumycetoma. Either of them can be acquired by walking barefoot. Which of the following is true about Cryptococcus? Pathogenic species are urease positive. Cryptococcus gatti is associated with bird guano. Extrapulmonary cryptococcosis is an AIDS-defining illness. Opportunistic infection is seen in AIDS when CD4 counts fall below 500. Now, please remember, I have taught this in my lectures also that cryptococci are urease positive. Okay, in fact, all cryptococci, whether they be pathogenic or non-pathogenic, are urease positive. Cryptococcus gatti is not associated with bird guano. It is associated with eucalyptus trees, soil around eucalyptus trees, right? It is Cryptococcus neoformans which is associated with soil contaminated with bird excreta or bird droppings or bird guano, right? So this statement is incorrect. Extrapulmonary cryptococcosis is an AIDS-defining illness. Definitely yes. Cryptococcal meningitis is an AIDS-defining illness. So this is correct. And opportunistic infection in AIDS occur when CD4 counts fall below not 500 but below 100. Right? So option A and C are correct. Our answer is option B. Okay. Remember, there are two pathogenic species of Cryptococcus. One is neoformans, which is capsular serotypes A and B. The other one is Cryptococcus gatti, which is capsular serotypes B and C, right? So it is gatti which is associated with eucalyptus trees, not bird guano. A 2-year-old child. Now here I'm not sure whether it was a 2-day-old, 2-month-old or a 2-year-old child. But whatever it is, let's see, read the question. Presents with watery diarrhea. Whenever we have a child less than 5 years of age, the first etiological agent which can cause diarrhea is rotavirus. Okay. On examination, the baby is severely dehydrated, which is the most likely cause. No doubts, it is rotavirus. Yes, E. coli, especially the enteropathogenic E. coli, can cause infantile diarrheas, but that is amongst bacteria, it is the most common cause of infantile diarrhea. Here, we are asked what is the most common cause of diarrhea in children. Undoubtedly, it is going to be rotavirus. A child, remember what is the morphology of rotaviruses? They have a wheel-shaped morphology. A child is brought to the emergency with complaints of diarrhea for last 3 days. Now there is blood in urine. Investigations reveal thrombocytopenia. I am not sure whether there was also mentioned something like anemia mentioned here, because this is diarrhea complicated with hemolytic uremic syndrome. How did we arrive at this? Because the patient has hematuria and the patient has thrombocytopenia, right? So HUS complicating the diarrhea in the child. What is the most common cause? Out here the options amongst, in fact, I am not sure whether it was E. coli 0157 H7 mentioned or it was enterohemorrhagic E. coli that was mentioned. It would be very nice of you if you could please put in your inputs in the comments of this video if I have made any mistakes in the wording of the questions or in the options. Grateful for that contribution, right? So our answer here is E. coli 0157 H7, right? So this is diarrhea complicated by hemolytic uremic syndrome. A male patient presents with urethritis. It can be caused by all except Trichomonas vaginalis, Gardnerella vaginalis, Mycoplasma genitalium, Neisseria gonorrhoeae. The most common cause of urethritis is Chlamydia trachomatis, serovars D2K. This is not even mentioned in the options. Neisseria gonorrhoeae can cause urethritis. Mycoplasma genitalium can cause. Trichomonas vaginalis can cause urethritis. However, Gardnerella vaginalis causes bacterial vaginosis, not urethritis. Okay, so that is our answer. Wrong about Ascaris lumbricoides. Massive infection can cause rectal prolapse. Most infections are asymptomatic. Eggs in stools are non-infective and need time in soil to become infective. Asthma-like conditions can be seen following infection. Now here the answer is option A. That is incorrect about Ascaris lumbricoides. Why is it incorrect? Ascaris lumbricoides, or the common round worm, the longest nematode found in the human intestines, resides in the small intestines, not in the large intestines. So it cannot cause rectal prolapse. Rectal prolapse is caused by a massive infection by Trichuris trichura or the whipworm, okay, but not by the common roundworm. Now these infections of common roundworm, most infections are asymptomatic, right? And so patient keeps on passing these eggs in stool. What are these two types of eggs? Fertilized eggs and unfertilized eggs. The fertilized eggs, these are containing an unsegmented ovum. That means they are not infective to man. Once they are passed out in stool, they need to mature in the soil so that the filariform larva can develop within it, and then it will become infective to man. Okay, so that statement was correct, that they are not infected when passed out in stool. Okay, and do they cause asthma-like symptoms? What is that syndrome called? It's called as Loeffler's syndrome. So once the eggs are ingested by humans, the filariform larva is released. It penetrates the GI mucosa, enters the portal circulation, reaches the right side of the heart, then passes through the lungs. During its passage through the lungs, it precipitates asthma-like symptoms, which is called as Loeffler syndrome, right? Then once they reach the lungs, they climb the respiratory tract, and then they are swallowed, and finally they mature as adult roundworms in the small intestines. Okay, most common cause of Loeffler syndrome is what we have been asked about, Ascaris lumbricoides, right? So the only statement that was incorrect was massive infection can cause rectal prolapse. That is seen with whipworm. A patient presents with abdominal pain. On examination, there is a mass palpable in the right hypochondrium. Ultrasound showed a cystic lesion in the liver. Ultrasound guided aspiration was done and microscopy of the aspirate is shown. What is the most likely infection? Tinea solium, Echinococcus granulosus, Hymenolapis nana, Tinea sandigenata. So we have a patient who has come with pain and lump in the hypochondrium. There is a cystic lesion seen in the liver. What is the first thing that should strike us regarding infections? It is Hydatid disease or Hydatid cyst. Okay. Ideally, we do not aspirate hydatid cyst because sometimes release of the hydatid fluid can cause anaphylactic shock, right? But here this examiner has told us that we have done ultrasound guided aspiration and we are seeing this kind of head-like structure and we are seeing these hooklets. So our protrusion, which is called as a rostellum with hooklets, this is just for confusing us. Our diagnosis is hydatid cyst. that is caused by Echinococcus granulosus. Remember, Echinococcus granulosus is one of the cestodes in which humans are never the definitive host. This is Echinococcus granulosus, that is the head with the rostellum and the hooklets. Then there is a neck. Then there are just 3 segments: immature, mature, and gravid segments. And where is the adult worm of Echinococcus found? If not in humans, it is found in the small intestines of dogs. So dog is the definitive host of Echinococcus granulosus. Accidental ingestion of eggs passed out in dog feces by humans, oncosphere is released, it penetrates the GI mucosa and it forms the hydatid cyst, most often in the liver. Okay, and this is what is the morphology of the hydatid cyst. There is a pericyst, an ectocyst and an endocyst, and from the endocyst, which is the germinal layer, we are seeing these brood capsules which have these scolices, and that was what was shown in the image. The scolex, that is the head with rostellum and hooklets. Which of the following is not involved in development of the naive T cell to an activated helper T cell? So we've been asked not involved. T cell interaction with antigen presenting cell, interaction between CD28 and CD28 ligand, PD1 on T cell interaction with PDL1 ligand on antigen presenting cell, cytokine mediated proliferation and differentiation. If you have studied or gone through my videos on immunology, which are slightly on the difficult side, you would have, of course, this question has been asked in other formats in INI-CET twice or thrice in the last 4 to 5 years. You can easily answer this question. Even if you've not heard of PD1 or PDL1, you know, naive helper T cells are continuously circulating in our body looking for antigens to recognize. Naive means non-activated, never yet interacted with an antigen. And when dendritic cells, which are the classical antigen presenting cells, take up an antigen, right, from any of the tissues, they are going to process them, they are going to create small peptides of them, and they are going to present them in the groove of the MHC class 2 and migrate to the regional lymph nodes, right? And waiting for a helper T cell, come and recognize this peptide that I'm presenting in the groove of my MHC class 2 antigen. The naive helper T cells will come and enter that lymph node where the dendritic cell is waiting. They will scan the surface of the dendritic cells. Do I recognize the peptides with my T cell receptor? If it does not recognize, it passes by, bye-bye. And if it recognizes, it gets the signal 1. I have got my signal 1. I have recognized the antigen in the groove of this MHC class 2. So yes, naive helper T cell, the signal 1 is recognizing, that is interaction of the T helper cell with the dendritic cell. Then here, once the interaction has started, let's see this diagram closely. That's our MHC class 2 antigen. This is our antigen presenting cell. And that's the peptide being recognized by the T cell receptor of the helper T cell. Then the helper T cell says, give me my second signal. So it got its first signal. The second signal is the CD28 of the helper T cell binding to the CD28 ligand. Do you know what is this ligand called as? It is B7, B7.1 and B7.2, CD80 or CD86. These together are also called as CD28 ligand. This provides the signal 2 to the helper T cell. So this we've understood, okay? And what's the third signal? The antigen presenting cell is going to secrete certain cytokines. If the cytokine secreted by the antigen presenting cell is interleukin 12, if it secretes interleukin 12, this helper T cell is going to convert into a Th1 cell, okay? So these are the 3 signals which are required for activation of helper T cell. Interaction of the T cell receptor with the peptide being presented in the groove of the MHC class 2, CD28 binding to the CD28 ligand on the antigen presenting cell, and the cytokine being secreted by the antigen presenting cell, depending on what kind of antigen it is presenting. If it is presenting, it is secreting interleukin 12, the helper T cell will convert into activated Th1 cell. If the dendritic cell is going to secrete interleukin 4, this helper T cell is going to convert into a Th2 cell. And if it is going to secrete interleukin 1 or interleukin 6 and interleukin 22, this is going to convert into a Th17 cell. Okay, so that's why our answer was option C, that was binding between this interaction. So even if you don't know about this is a slightly higher level. These are basically inhibitory to helpers, inhibitory signal. Binding of PD1 on the T cell, binding to its ligand on the APC is actually an inhibitory signal, not an activation signal. Okay, let's move on to our last question. Which of the following, when administered to a patient, does not impart passive immunity? Monoclonal antibody, of course, you're giving preformed antibody, you're giving preformed immunoglobulin A, you're giving preformed antitoxin. These are all methods of imparting passive immunity. However, administration of toxoid, giving diphtheria toxoid or any other toxoid like tetanus toxoid, what does it going to act as? It is going to act as antigen, which is going to induce antibody in the patient who is vaccinated or administered the toxoid. So this is going to be active immunity. Okay, active, to be more specific, artificial active immunity. Right, so that is why our answer to this question is toxoid. That's all for our discussion. Bye-bye.